Hello, my name is David Kornspan. I'm from the Kimron Veterinary Institute in Israel. I'm really happy to attend this seminar. And here I would like to share you with our recent study regarding the acidic stress response of Brucella melitensis, new insights from a transcriptome analysis. Regarding the Brucella species, Brucella is a gram-negative intracellular pathogen that causes the zoonotic disease brucellosis, which causes abortions in its natural host. There are 12 known Brucella species, where the 4 which are the most pathogenic to humans include Brucella melitensis, which its natural host are small ruminants, Brucella abortus, which its natural host are cattle, Brucella suis, its natural host are pigs, and Brucella canis, which its natural host are dogs. Brucellosis is one of the most frequently encountered zoonotic diseases. There are around 500,000 new cases reported every year around the world. Humans can acquire infection by contact direct with infected animal or by consuming animal products, such as unpasteurized milk. If brucellosis is not treated well, it can cause in humans severe complications. Regarding the pathogenesis of Brucella do not produce the classical virulence factors, toxins and plasmids. Brucella is able to survive and multiply within various host cells. The major virulence factors of Brucella are the lipopolysaccharide, the type 4 secretion system, which allows the interaction with host cell surface, the formation of Brucella-containing vacuole, and their interaction with the ER, where the bacteria multiply. Brucella can modify immune response in host cells. They cause the enlargement of lymph nodes, liver, and spleen. And their pathogenicity is dependent on their ability to multiply and survive within macrophages. It is important to know that Brucella species must cope with various acidic environments. First, when they penetrate into the host via the gastrointestinal route, and also during the initial stages of the intracellular infection, where there it is required the acidification of the phagosomal compartment to a pH of around 4.5 before intracellular replication occurs. It is important to know that the requirement of this low pH is transient. It only extends through the initial stages of the intracellular infection. While this acidification causes the upregulation, the induction of the expression of the verb Operon, which controls the expression of the type 4 secretion system. This Operon, the verb Operon, interacts with the ER to neutralize the pH of the phagosome. There are two main acid-related response systems in bacteria. The first is called the acid resistance system, the AR. This is the response to extreme acidic stress. pH such as 1 to 3, it's the pH of the stomach. Bacteria use such as urease and repair mechanisms. The second system is the acid tolerance response, the ATR, to mild or moderate acidic stress. For this, bacteria use such as ATPase systems. Brucella must tolerate the strong acidic environment of the stomach. It was suggested that urease plays a significant role in protection of these bacteria under the highly acidic stress of the stomach. What we are interested in finding out was how do Brucella species cope with the acidic stress inside the Brucella containing vacuole, meaning how do Brucella cope with the pH of 4.5. A transcription-level regulation network of Brucella within the acidic environment has been recently reported. You can see here two reports, one in 2015 and 2016, showing an up or down regulation of specific genes in Brucella melitensis under acidic stressed conditions. What we have done, we have investigated the acid tolerance of the virulent Brucella melitensis strain 16M grown for 4 hours, either under neutral pH conditions or acidic pH conditions. We examine the expression of genes using RNA-seq. We integrated our data with that published in two models, one by Rossetti in a in cellulo cell culture model, and the second by Boggiato in an in vivo goat supermammary lymph node model. Our goal was to elucidate the specific mechanisms that potentially allow this pathogen to cope with the rapid decrease in pH within its host. In our analysis, we found 760 genes in Brucella melitensis strain 16M which were significantly differentially expressed in bacteria grown under neutral and acidic pH conditions. In this figure, you can see on the upper side our biological pathways of genes which were upregulated and associated to microbial metabolism and oxidative phosphorylation. And the bottom are biological pathways of genes which were downregulated and associated to metabolic pathways and ABC transporters. We explored protein-protein interaction networks within these 760 genes, and we found a network of 25 proteins, which 22 of them were related to oxidative phosphorylation processes. We then compared our data obtained by our analysis to that reported by an in silico cell model by Rossetti, which characterized the transcriptional profile of Brucella melitensis in a model of HeLa cells infected by Brucella melitensis at 2 points, at 4 hours post-infection and 12 hours post-infection. Our aim was to compare our genes, which were up or down regulated, to those which were up or down regulated in their model, in order to locate which are the genes which are differentially expressed in a pure acidic environment, but not in an in cellulo model. As you can see here, surprisingly, most genes were uniquely expressed in each group, while only a minority of genes were common in both lists. We then compared our data to the previously reported transcriptional profile in an in vivo infection model. Boggiato examined the transcriptional profile of Brucella melitensis 16M RNA obtained from the supermammary lymph node of experimental infected goats at 4 and 38 weeks post-infection. We aimed here to investigate the differences between the expression of genes in Brucella melitensis grown in pure acidic conditions compared to the in vivo model. As you can see here, that as in the in cellular model, also here in the in vivo model, most of the genes were unique to each model, and only 17 differentially expressed genes were common to all three conditions. You can see here a heat map demonstrating the unique expressional profile of Brucella melitensis under acidic conditions. We took our genes, which were differentially expressed, and you can see here that they are categorized into 4 clusters. Cluster 2 and 3 demonstrate the unique expressional profile of Brucella melitensis under acidic conditions. You can see here it's obvious the difference in the expression of the genes in these two clusters. The main biological pathways of the genes in these two clusters are metabolic pathways, ABC transporters, oxidative phosphorylation, cytochrome Bo oxidase electron transfer, and RND efflux pump and sulfur metabolism. We next aim to reveal the key genes in Brucella melitensis, which are differentially expressed specifically in response to pure acidic stress and were not differentially expressed in the other two models, the in vivo and in cellular model. We revealed 588 genes which were uniquely expressed in our model. Part were induced and part were repressed. When we annotated these genes, we revealed 48 genes which were involved in various important pathways associated to survival and virulence of this pathogen. 38 were upregulated and 10 were downregulated. Among these genes, we found 13 genes which were highly upregulated, and these key genes are associated to the bacterial response to acidic stress, and we assume that they play a vital role in the response of Brucella melitensis to acidic stress. Among these genes, we found 6 which were highly upregulated, and these genes belong to the Urease Operon. In several bacterial species, Urease was shown to play an important role in the bacterial acidic response by generating ammonia, which is protonated to ammonium, thereby consuming intracellular protons, causing increase in the intracellular pH. Most members of Brucella show strong urease activity. It has been suggested that this urease protects Brucella during the highly acidic environment in the stomach. The significant upregulation of 6 genes of the Urease Operon in Brucella melitensis under moderate acidic stress conditions suggests that Brucella may use this urease activity not only during extreme acidic stress, but also during the moderate pH of the Brucella containing vacuole in the initial stages of infection. Among the genes that were highly upregulated were 4 major facilitated superfamily efflux pumps. In several of pathogenic bacteria, it was shown that these efflux pumps play a highly important role in virulence and survival of the bacteria. It was shown that genes encoding efflux pumps in Djla were induced under acidic stress. Thereby, it is possible that similar to what happens in Djla, also Brucella melitensis upregulates these genes as a secondary mechanism to cope with acidic stress. Another interesting gene that was highly upregulated in Brucella melitensis under acidic stress was a gene encoding the molecular chaperone Djla. It is known that molecular chaperones facilitate protein folding and prevent protein denaturation and are involved in various cellular processes. Under acidic conditions, partly unfolded proteins may emerge. Thereby, these chaperones may stabilize them to prevent acid-induced aggregation. In Legionella, it has been shown that the Djla chaperone plays an important role in intercellular growth and also in coping with various stress such as pH acidic stress. The upregulation of this gene coding the Djla in Brucella melitensis under acidic stress may also play an important role in preventing irreversible aggregation of misfolded proteins, contributing to bacterial survival within its host. We have found a significant upregulation of 3 genes encoding type 4 secretion system proteins VERB1, 3, and 4. The VERB type 4 secretion systems consist of 12 genes which play an important role in mediating intracellular survival and manipulating the host immune response to infection. It is known that induction of the VERB type 4 secretion system requires the acidification of the Brucella containing vacuole. Thereby, the highly significant upregulation of VERB1, 3, and 4 by Brucella melitensis under acidic stress strengthens the previous reports regarding the importance of these genes and their survival of Brucella within the harsh intracellular environment of its host. We also found a significant downregulation of 5 key genes which encode proteins that participate in critical stages of the bacterial cell cycle. 2 DNA translocation proteins and 3 cell division proteins. This finding may indicate that under acidic stress, Brucella melitensis may arrest cell division and activate pathways to bypass or repair damaged DNA. This mechanism is known as the SOS response, and support for this conclusion is from what we found a highly upregulation of 3 genes encoding nucleases and play an important role in DNA repair processes. The conclusions of our study, after conducting a comparative transcriptomic analysis of Brucella melitensis grown either under acidic or neutral pH conditions, comparing our analysis to that published before in 2 different models, the in cellulo and in vivo model, we found several genes that play key roles in various crucial pathways of Brucella that were either up or down regulated under acidic stress. We suggest that these genes are specifically involved in the molecular mechanisms underlying the Brucella melitensis response to acidic environments. Our further plans are to conduct mutation and knockout experiments in order to determine the specific role of these genes in acid resistance. Thank you for listening. I want to thank you for honoring me with the opportunity to participate in this important meeting. I want to thank the people I work with, Professor Mari Salmon-Divon from Ariel University, who has conducted all the bioinformatic work and the analysis, and we work together on this big project. I want to thank the people I work with in the Kimron Veterinary Institute, Dr. Shlomo Blum and Dr. Svetlana Bardenstein. Thank you and goodbye.